Thank you very much. Can everyone hear me? I'm really very honored to be able to come here and be able to interact with some of the grad students. We went out to dinner and had a good time. I probably ordered the wrong stuff, some hot food, but that was okay. I actually have a fair amount of connection with Cornell people in the past. I think particularly those of you that are graduate students ought to realize how fortunate you are to be able to work at an institution which has a lot of depth in the different areas of plant biology, and just the people that are your mentors. They're well-known people all over the world, and in my interactions throughout my career, I've met so many people, and I find, oh my gosh, he's from Cornell, she's from Cornell. You know, Cornell has a reputation for graduating a lot of people. I realize there's a lot of molecular work and bigger focus on the molecular end of things. But what I've noticed over my career is that a lot of the people I've interacted with have good basic knowledge, but they're also able to reach out to the growers. And of course, I do a lot of very applied work, and so I have a lot of interaction with vegetable growers, and there's just a number of people from Cornell that I've seen over the years interact well and give a lot of information. They take the basic information and bring it down to the growers level, and this will always continue to be important. Regardless of the balance of research that's going on, the information in agriculture at some point has to get to the growers. And I think some institutions lose sight of this. And there's been several people from Cornell that have given me some assistance through the years. Molly John, Henry Munger, who passed away some years back. Tom Zitter, pathologist, has helped me out in sending me cultures and diagnosing certain disease problems that we had, and just a number of people. And I've also met a lot of people. I used to work in agricultural plastics a lot, and it turns out that probably because of the particular expertise at Cornell University, a lot of those people were trained at Cornell University. And so anyway, this is a real honor to be able to speak to you, and we'll get started here. So Cucurbita, I do melon breeding, but I'm not going to talk about that at all. Cucurbita is an extremely interesting crop, and all the species originated in the Americas, in North and South America. Some of them at an extremely early date. And to start off with, C. maxima on the bottom originated, and we know the wild species, it's the progenitor of C. maxima. And the interesting thing is the distribution of this particular species by the time the Europeans colonized the Americas, the distribution was probably about like that, and it wasn't up in the United States or Central America, and we don't have a lot of archaeological sites where they've recovered maxima. It goes back maybe 3,000 or 4,000 or 5,000 years ago, the domestication, but it's one of the younger species. Ficifolia, I'm not going to talk about. It grows at high altitude. It will not intercross with any of the other domesticated species. It has very little variation. It's almost monotypic, but it's adapted to very high elevations and has not found a lot of worldwide use. Cucurbita Maxima is the one species that's most adapted to the tropics, and that's probably affected how it was distributed. It was originally thought to have originated in Mexico, because there were archaeological dates for its existence going back like 6,000 years ago. But now they have samples that they've been able to get from sites in this area that go back about 9 to 10,000 years ago. And it's usually in these archaeological sites, the material that's preserved are seeds and fruits, pieces of fruit rind, but it's usually the seeds that characterize the particular variety. If we have the stem of the fruit, the peduncle of the fruit, we can also characterize varieties that way. And so it is thought that it originated here because there's a lot of variation and a lot of very relic varieties that exist in Colombia. I'm not going to talk about a gyrosperma. It's mainly used for its seeds, and it hasn't been utilized much throughout the world. Cucurbita pepo, subspecies pepo, originated in this area, and then again, it goes back at least 9,000 years, and there's been several archaeological sites where they found this particular species. And there's another subspecies of pepo ovifera, and it's quite interesting that these two species, the wild species from which ovifera came, exist down in this area or up into this area. And so we know the wild progenitor of this particular species, and the Native Americans introduced and bred this species going back about 5,000 years ago, and it spread throughout the eastern half of the U.S., not so much over in the western half of the U.S. Whereas this species originated, and there's archaeological sites down in southern Mexico and also along in this area, the state of Tamaulipas in Mexico. And the most interesting thing is even though these species are easily crossable, you know, you could almost consider them one species, but there was no contact. We don't have any knowledge of contact prior to the Europeans coming that these species ever crossed with each other. So that it's very unusual to find two species that were domesticated so close to one another. And yes, there apparently was not a lot of human contact, and so there wasn't a lot of intercrossing of these particular species. So the variation in this species is well documented, and so I'm going to more talk about the variation that relates to my breeding programs, but you can go back into herbals back into the 16th century and see quite a bit of variation in the species, because the Europeans, when they came over and conquered some of the countries, they brought back the fruit to Europe. And Duchesne's drawings in 1786, Natural History of the Gourds, it shows about all the phenotypes that exist today in Cucurbita pepo subspecies ovifera. And excuse me. And then the vegetable garden is kind of interesting to look at it because it shows the state of the variation in cucurbits and other vegetables existing in the 19th century. And then the cucurbits vegetables in New York, that's always there's a whole series of vegetables of New York that was published back then. And I have copies of several of the vegetables, and they're just fun to look at. And there's a whole lot of books. The Complete Squash by Amy Goldman is probably one of the best of them. This is what was in my greenhouse 3 weeks ago that came from the field. And I would just have to tell you that all the variation that you see in all those catalogs and stuff, a good share of it exists in this particular photo. I work with all three species and the two subspecies, and it's almost overwhelming. It's a very daunting task for me at this stage in my career. And these all involve selections. And we have to do cooking tests and various evaluations on all these squash in these two pictures. So Cucurbita Maxima, just to refresh your interest or refresh your knowledge of it, the main Cucurbita Maxima that you see in supermarkets is kabocha or buttercup. We generally call the squash buttercup if it has a protruding ovary, whereas in the Asian countries, they've gotten rid of that and it's called kabocha squash, but that's a general term in Asia for any small squash that looks like this. And so the processing, Golden Delicious is the main processing squash. There are a few hybrids, but mostly just open pollinated. And actually in Argentina, you have summer squash produced. They let it grow a little bigger than our summer squash in order to get big enough, but they do use it as a summer squash. And then there's several others that I can't show, but the giant show pumpkin are really an anomaly. The size is just unbelievable in those. And then if we go to Cucurbita people, subspecies ovifera, the Acorn Group, even though you probably don't think of this as an acorn, this is a delicata type and a sweet dumpling type, but it's all called the Acorn Group. In some books, the straight necks and the crook necks, summer squash, and the scallops are all put into separate groups. I call them all summer squash, a squash that's eaten immature. And I know for some reason in New England and the Northeast, a zucchini is a zucchini and a summer squash is a yellow squash. They're all summer squash. they're all eaten immature, okay? And then gourds, I define gourds as squash having a hard shell and having some ornamental value. And that's not always the definition getting. And then here's what I mean about Cucurbita people, subspecies people, the one that originated in Mexico. For one thing, the size is bigger, and you have some that are quite large. You have quite a lot of variation in color. These are some of my breeding lines. I saw in a store window when we were eating last night, I saw a lot of variation, looked just like this, matter of fact, but it was all fake pumpkins. It was plastic pumpkin. It's kind of discouraging, actually. And then in the summer squash arena, this is probably the dominant use of squash is for summer squash, particularly the zucchini types, and they were derived from the cocazelle from Italy, and these types were bred actually in Europe. As far as we know, they were not bred in the area of origin. In Cucurbita Masada, I didn't start working with Cucurbita Masada until just really the last 12 years or so. But I had seed companies come to me in the 80s, particularly Libby's was breeding a processing, this Dickinson field processing type, and they sent me some lines to look at, and I looked at them. They're really poor quality for edibility, and I thought, well, I'm not gonna work on Cucurbita Masada. It's butternuts and it's this Dickinson field type, and the seed companies, they can do all the work on that. I'm not going to get involved with that. Well, now it's my biggest breeding project, you know, and I'll tell you why in a little bit, why I'm so excited about it. And if you go into third world countries, because we've adapted some of these varieties so they grow in temperate climates, and some of these, but the calabaza that exists down in the Caribbean, and there's a lot of varieties of Masada that grow in Southeast Asia. And if you go to India, and all the squash in the market, I don't have a picture of one here, but later on, there's sort of the large cheese type. You see them in the market. Not the type of quality we would think for you know, like a butternut, but nonetheless, they're very popular in Asian countries. The fruit of squash is a pepo. Technically, it's a modified berry, and the flesh actually is derived both from the ovary and it's also derived from the floral cup or the receptacle, and which makes it interesting because occasionally you get a selection and there'll be two colors. There'll be a real intense color in the ovary part and also in the placental material, and then this outer ring of flesh will be totally a different color. I couldn't find a good slide of that to show you, because you could have gene expression for carotenoids. You could have different expression in the receptacle tissue versus the ovary tissue. Kind of fascinating. It's a monoecious flowering species, bee pollinated. Bumblebees are the most efficient pollinators. There's also a cucurbit bee that actually evolved with squash. It needs to have open soil in order to nest, and so we don't have very many of the squash bees at the farm. They're not quite as efficient as bumblebees, and also honeybees will pollinate it. And so I'm not going to go into the intricate structure. The main thing is it's monoecious, separate male and female flowers, and they flower in the morning, okay? Now, it'd make a beautiful ornamental plant. We have flowers that are that big. I'm not exaggerating. In cucurbita people, we have some that are a foot in diameter. Beautiful flowers, but they wilt during the morning, okay? And so in that sense, you couldn't bring them in the house and use them as an ornamental. So this just shows the different species, and they have slightly different flower structures. And when you work with them, you can kind of see that this is kind of a bell-shaped, and this has the very deep lobes in it, and the Masada is a little bit different. The thing from a breeding standpoint, what one needs to know is these flowers wilt down at different times. It's genetically endowed in them. So the cucurbita people, it often wilts down by 9 in the floor. And by wilts down, I mean the flower loses its turgor. And when it loses its turgor in the male flower, the pollen loses its ability to germinate on the stigma. So you have to pollinate in the morning, okay? And so it turns out that the subspecies ovifera is the first to wilt down. And so we have to pollinate it by 8 or 8:30. Then we move over to this field, and it's got the people, and we can pollinate it depending on how cloudy or sunny it is. We can pollinate it late. And then the Masadas we pollinate between 9:30 and 10:30, and then we move over to the Maximus. It's the only way with a small crew that we can deal with all these species and get our work done. And we have to go in the day before and we tie these flowers off, and you can tell when they're going to flower the next day. It is possible you can take a female flower that's at this stage and you can pollinate it, and it will set fine. So you do have a little bit of leeway. You can also, the male pollen, even before it's shed, can fertilize, but it's kind of difficult to get the pollen out. So normally we like to have that flower open. So if you look at the growth curve, and this happens to be a seed pumpkin example, which is a little bit extreme because most of the energy is going into the seeds rather than in the flesh. But the pumpkins and squash behave like any crop that produces a storage organ, like potatoes and so forth. In other words, the vegetative growth peaks usually shortly after flowering, and then as the leaves senesce, it goes downhill. And then you have in the pericarp, in the flesh of the pumpkin, you have the biomass is mainly going into the flesh here, but it's also going into the seeds. So in the peak dry matter of the flesh occurs earlier than it does with the seed. And so what actually happens is if you get too much plant senescence, the seed is still developing in that fruit, and it actually takes away starch reserves from the flesh. you know, and it's important to know, because if you pick your fruit too early in development, then you're interested in the flesh, not the seeds. You're going to have respiration occurring, and you're also going to have transport of sugars from the flesh, you know, starch breakdown and transport into the seeds. So it's important to have the knowledge of the growth cycle. And this is just another one. It's more detailed work that we did, graduate student did back in the late 90s, and actually looked at embryo growth, because no one had looked at embryo growth. And if I should have had a picture of a fruit cut open, if you cut open a fruit at 20 days after fruit set or after pollination, if you look at that fruit, you'll say, oh, the seeds are fully developed. Well, what it is, the seed coat's fully developed. Not fully developed, it reaches its full size. And the seed size, you don't see segregation for seed size in a fruit. It's a maternal trait, and it's determined by the seed coat, which is maternal tissue. So that's why all the seeds, even if you have segregation for seed size, you don't see it until the next generation, okay? And so what happens if you opened up that seed at 20 days, I should have put in a picture of that, but the embryo is very small, only a couple of millimeters big. And so the embryo reaches near full size in the next 20 days, but it actually accumulates lipids and proteins up to 55 to 60 days. And so it's important to know. The actual fruit growth, if you look at the fruit growth curves, depends on the size of the fruit. If it's a small gourd, actually the fruit reaches maximum size in about 12 days. And some of the acorn squash, 15 to 18 days. And in like a pumpkin, a 25-pound pumpkin, will reach its full size in about 25 days. If you're talking about the show pumpkins, and the only reason I know this is I've given talks to these growers. They're fanatics about growing these big pumpkins. Retired farmers, that's what they do. They grow the show pumpkins. And so one had a nice growth curve of a show pumpkin. He measured circumference over time. I was really amazed. These show pumpkins are still growing in girth at 60 days. At 60 days. It's pretty amazing. Quite a different curve from this curve. And again, the mesocarp dry weight, the starch content peaks really close to 30 days, 35 days. And then it usually goes down. Again, this is with a seed pumpkin. And so again, in the case of the seed pumpkin, it needs mesocarp. It needs to get sugars from the mesocarp. So the mesocarp goes down a little in order to feed the embryo growth. And when I talk about pumpkins to growers, this is just a chart I would show pumpkin growers, because it's a little different. In a pumpkin, all you need is an orange pumpkin, right? And so that color change usually occurs between 25 and 40 days after pollination. There's some varieties that are a little later, but most varieties, some of them reach full color by maybe 32 days after pollination. Well, can you harvest? The grower always asks, well, can I harvest my pumpkins right after they turn orange? I said, yeah, go ahead. You know, and if you have a healthy plant, the peduncle will be fine. The stem of the pumpkin, that's in breeding pumpkins, that's the most important part. If you don't have a good stem, if you can't carry a pumpkin out of the field on its stem, it's not good. And so it turns out that cellulose and lignin, most of that is accumulated in the peduncle, most of it by 30 days, and there's a difference in varieties on that. And so, and again, we actually did a study of peduncle dry matter and dry weight in pumpkin, because you have to know that, okay, in order to know when to harvest it. But of course, the seed's not mature by then. So in my work, my timeline for my cucurbit breeding projects at UNH, I was breeding melons all since I started. I won't talk about melons, but I started off, I was only working with Cucurbita Maxima for a number of years. And I'll be honest with you, my first variety in Maxima came out in 1981. Well, that's 13 years of breeding to produce my first variety. That doesn't say too much maybe about my breeding expertise. It was a very difficult project. I was trying to put bush genes into a large fruited processing pumpkin because there was a huge processing industry in New England and the Northeast at the time in the golden delicious type. And I had a particular ideal type of pumpkin that I envisioned. A more upright bush pumpkin would be more efficient photosynthetically. And we did a lot of background work, you know, comparing the phenotype of bush and the leaf area and light interception and so forth. I'm not going to go into that, but it was a pretty difficult project. Well, in the lines you see that are in dark, these are the projects where I've spent the most research time on, but they're not the projects that, from a standpoint of getting something into the industry, were successful. And that, I think that's how it is at an institution. You work on some projects that seed companies wouldn't necessarily work on, but maybe those will never be successful at getting something into the seed industry. So with me, because I like to breed an end product, I tried to have enough projects so I could introduce varieties into the seed market, you know, so the growers would think I was actually doing something at UNH, you know. If you're a breeder and you're there for 13 years and you don't have much to show for it, and you go to a grower meeting, you're giving a talk, and they're shaking their heads, you know, what the hell's Lloyd doing? You know, I mean, that's always bothered me. So what happened to this bush strain? Well, Agway tried to introduce it and so forth. Well, the processing companies, they're back in the 19th century, you know, in terms of how they space things. And so they were used to growing, processing pumpkins 12 by 12, and that was their cultivation system. It doesn't matter what we told them. I could tell them, look, you need high density planting. You need to plant this bush strain, you know, close spacing. I give them the spacing, but you go out in their fields, it was 12 by 12. And they were telling me, well, it doesn't yield. Well, no, a bush plant that's 3 to 4 feet in diameter at 12-foot spacing is not going to give a very good yield, is it? You know, so what happened in New England? By the time I got my first variety, the processing industry was gone. I mean, just gone, left completely by the mid 1985. Well, I continued these lines, processing lines. I had them, and it turns out now that I'm actually using some of those processing lines for breeding rootstocks for melon. Okay, so I'm glad I kind of hung on to those. But and the same thing, I use the bush genes that I've accumulated, because it's not just one bush gene, as you see in the literature. Actually, in Maxima, we've identified 3 bush genes, and they're all incompletely dominant, so difficult to work with. And so we have developed a number of bush lines, high quality. Again, all our material was very low quality, you know, in terms of starch content. And the genes for increasing starch content, it's a quantitative trait, and somewhat difficult. Hulless pumpkins, I started the work and was doing a lot of work on, you know, well, what's the composition of the hull and this and that, and had a fair number of graduate students work on hulless seeded pumpkin. Well, it's not an easy industry to get going in the U.S., North America. A lot of the snack food companies, trail mix companies say they would like North America, they'd like seed available from North America. They relied on China. Okay, there was a down year in China 2 years ago. They didn't produce enough seed. So all these trail mix companies were crying for seed, and they're all saying, well, we want some North America growers. But what they don't want, and they're not willing to have contracts. You can't ask a grower to come up with $1 million for the harvesters, the seed cleaners, the seed dryers, and all you need for this crop without having long-term contracts. And so I do have a few varieties coming out, and working with a processing company in Illinois now on the hulless pumpkin. So maybe something will come of that. Ornamental pumpkins, pretty easy to breed. You know, I can't claim I was a genius working with these. I've got a lot of varieties, probably 30 varieties or so have my germplasm in. you know, it's just one of those things, you know, where it's squash, you know, I think I got a total of 9 varieties of squash, you know, but I've spent a lot of time breeding squash. And so, and this, for some reason, my computer just won't, my upgraded computer won't accept the changes. I've changed this table a million times. Anyway, the gist of it is, when I came in 1967, these are the number of genes that were described for these 3 species, if you looked in the literature. Now, maybe I might have missed a few in the literature, but pretty much this is it. And half of them were described wrongly. You know, they had the wrong inheritance model for them or whatever. Okay, and in 2017, these are just genes for morphological traits. Okay, morphological traits. Okay, it's not including the genes for disease resistance. But even 2017, approximately 20 genes in Cucurbita Maxima that you can trace to morphological traits, 39 in people and 9 in Masada. That is really not much at all. Okay, most of the genetics I work with, I'm not going to say most of it. I mean, I work with a lot of those genes, but there's so much variation that I work with. There's no genetic information on it except what's in my breeding book. And so, as a breeder, you've got 2 directions you could go. You know, if I find 5 traits, I can either spend all my time, you know, producing isogenic lines and making the crosses, you know, to publish an inheritance study, or I can release some varieties to grow. I've made the selfish choice, perhaps, to breed varieties for the growers, and maybe I'll write a book, small book sometime, just detailing, you know, kind of what I know about these descriptive morphological traits. And I go back to my first breeding course was when I was an undergraduate at Oklahoma State, and this chart in an old breeding book by Allard, really struck me, you know, because you see all this variation in plants, and I was actually doing special projects for a couple breeders at Oklahoma State at the time, and I'm seeing, oh my gosh, if you had 4 recessive genes in a perfect population, this was like your Punnett square perfect population, only 1 out of 256 plants would have all 4 of those genes homozygous, okay? In other words, true breeding. That's just with four genes. Now you're in much better shape. You know, about a third, if it was all dominant genes, about a third of the plants would at least have at least 1 dominant gene in it, you know. And if you're lucky enough to work with traits that have incomplete dominance, you're a little better off because you can see the recessive phenotype, the dominant phenotype, and the intermediate phenotype. But in squash, this is important because your population size is limited. The biggest population I ever used in squash was 450 plants. 450 plants. A wheat breeder would laugh at that. It was a wheat breeder that was teaching this course I was taking, you know. Of course, he was talking about populations, probably, you know, F2s of 3,000 wheat plants or something, you know. Well, you know, in an acre of squash breeding where we have guard rows and different plots and so forth, you're talking 6 to 9, and whether it's bush or vine, you're talking 600 to 900 plants, you know, could be per acre is all you can grow, you know, and you're not going to devote all that to just 1 cross, and so it's quite restrictive. And remember, it's not self-pollinating species, so any plant that we save for the next generation the flowers have to be tied off. You do your self-pollinations, and then that continues the next generation, okay? And you can occasionally, like in summer squash particularly, if you didn't get a self-pollination and you've already made your, on summer squash, you're already selecting what you need, you know, by the time you pollinate it, because you're interested in the immature fruit. Then you could make cuttings and take those to the greenhouse. And occasionally we have taken cuttings late in the season, but often you can't. As soon as the growing point starts to senesce, you can get your cutting to root, but we've tried different hormones and everything. We can't get the growing point. We could do it in tissue culture, but we're not willing to go there. The same way, this is just it's a simplified example, but it kind of shows what you're up against. A lot of the traits we work with, like starch content and seed size and fruit size to some extent, are essentially what we call additive traits. If they're quantitative, we call them additive. If they're more qualitative traits, we say incomplete dominance. But essentially, you're talking about where your small a here is like adds 0 increment of growth, this 1 increment of growth, 2 increment of growth. Of course, the different genes, you know, don't all affect the trait in the same way. But the point I want to make here is if you have a quantity trait that has additive genetic variation, like in the case of starch content, it works out very nice because you know that when you get your highest starch content in a population, you're likely to have a lot of homozygosity. And this is what we found. If you're breeding a processor where you want very low dry matter content, you might be down at this end. If you try to select if you make a wide cross with something high starch and low starch, then in here, you're going to get some homozygosity, some homozygous lines, and some of them, you can have different combinations which would give you the same starch content. But you have a lot of heterozygosity, makes selection very difficult, okay? And remember, you're dealing with small populations. Another aspect that's important if I'm dealing with all 3 species is sometimes it's desirable to transfer traits between these species. Well, forget Maxima times people, okay? Very difficult to transfer genes. There's just too much sterility. I tried this early on and didn't have any luck. And actually, there were some traits I wanted to transfer between Cucurbita people, acorn squash, and butternut squash. I tried for 5 years to make that cross and couldn't get it to work. I'll tell you in a second how I finally got it to work, but had to use embryo culture. Cucurbita Maxima and Masada, if Maxima is the female, it's often you can get very good seed yields in this cross, but the F1 is sterile, okay? And so there's a lot of sterility barriers in the next generation. Usually you can back cross using your F1 as a female, you can back cross, but the other point I want to make is it's not cut and dried. You can't just say, well, you can cross Maxima to Masada. No, I could take 20 lines and I could make this cross. Some of them would cross very well. Some of them wouldn't cross at all. Some of them give some fertility. And basically, with what we know, maybe when we get molecular markers and we understand the genes that control this, fine, we could find the parents that are going to cross. But it's just a hit and miss, you know, situation. You know, we were fortunate in Maxima that one of my bush processors combines very well with Masada. Virtually all the different Masada strains cross well with this one strain of C. Maxima, but it's not always to do. And I told you I couldn't cross acorn in Masada, but I was able to make a cross of acorn and pumpkin, which is in the other subspecies, and I was able to cross that to Masada, and I got 7 seeds. We were able to culture those and get 5 plants out of it, and with those 5 plants, I was able to back cross in either direction. And so why were we interested in that? Well, acorn squash have very low carotenoid content. Carotenoid is one of the key nutrients in squash. And so this is just work we've just been doing lately because I just started working with interspecies hybrids. But anyway, we think this is much higher carotenoid levels. Both of these have much higher carotenoid levels than we have in acorn squash. Probably this one is probably, I'd say 3 or 4 times what we usually see in acorn. So it really would upgrade the nutrition if we could get this level of carotenoid into the acorn squash, which is an important squash. Acorn, butternut, and buttercup, kabocha, those are the 3 major squash in supermarket. So we took the same squash and crossed to Masada and got enough seeds so we could grow out 15 plants, and those plants segregated for pretty intense carotenoid content, and we're able to cross that with acorn. We've only got a few seeds, partial embryos. We're hoping this winter that we can culture those, do an embryo culture and recover plants to continue that project. My actually first interest in these interspecies hybrids was to make crosses between Maxima Golden Delicious and a processing type like large cheese or Dickinson Field. And we wanted to see what the yield would be in the F1. And the reason for that is that we had a hypothesis that because these F1 hybrids are sterile, they don't produce seeds, that we could increase yield because all the photosynthate energy could go to the flesh and not the seed. And sure enough, and we got a nice phenotype. It's semi-bush because we're crossing bush times vine, and the yields were really quite staggering compared to our Czech, which was a Dickinson Field type I got from Roop Seeds. The dry matter yield was almost double in this particular interspecies hybrid. Some of them don't work at all. Some of them produce misshapen fruit. Another avenue of research, because the interspecies hybrid, even if we get higher yields, the processor is not likely to adopt it because of difficulties in seed production. But so we've been using some of the interspecies hybrids as rootstocks for melon to overcome vine decline that occurs throughout the Northeast in melon. And sure enough, we can overcome that soil-borne disease quite easily on rootstocks. We can increase yield by 50% and also extend the growing season. We think this could be important in the future. Well, there's not a lot of melon acreage in New England, but still we're the seed production is again expensive. And one of the things we've been able to do is we've been able to take some of these intermediate Maxima strains and cross them to butternut. And originally the idea of this, we wanted to get a butternut shape in Maxima. And we just haven't been able to do that. We just haven't been able to transfer those genes back to Maxima. But the interesting thing is, through a series of crosses, we did end up with pretty much standard butternut. And that's only after a few back crosses, 3 back crosses to Masada. We essentially have a butternut type. What's the advantage of this? The seed fertility is 100%. So if we can take this and cross it back to Maxima, and we can get 100% of well-filled seeds, you know, and if it's compatible with melon and gives the same melon yields and disease resistance, then it could be a major breakthrough in the industry. Because normally when you make these crosses, a good share of the seed is not filled. You got to do, and even with commercial seed that we've tried, you do the grafting studies and a lot of the seed doesn't germinate uniformly. I'm going to have to go through this real quick. I'm running out of, okay. But another area of variation that we look for is in carbohydrate source, the balance of sugars and starch. That's what really determines whether a squash tastes good or is edible. There's other factors, but sugars and starch totally dominate the edibility of squash. And then in terms of vitamin content, of course, the carotenoids are, and I'm going to go through this because I'm running out of time. But the thing I want to show you is it's very expensive to measure starch content, okay? It takes a lot of extractions. Even if you send it off, it's very expensive, and you need to send a lot of material to be analyzed. And so we don't measure starch, but the interesting thing is starch is correlated very strongly at harvest with the percent dry weight. And we know that if you have a percent dry matter in the squash up around 18 to 20%, your starch content is going to be around 8 or 9%. And if you have a squash with that starch content, it has a nice pasty texture, a very desirable texture, and it's enough starch to produce high sugar content. So it results in a good balance of sugar and starch. The interesting thing is in the commercial varieties, they fall in the two most popular or three most popular commercial varieties. They fall into dry matter contents in all our trials. They average about 11%. And I can go to a supermarket in the wintertime or most any time, and I can test acorn squash from a supermarket, not kabocha squash or butternut, but acorn squash, and they test out between 9 and 11%. Essentially, we would call these inedible. And so, again, we just get percent dry matter. So we just get these small samples, dry them down, determine percent dry matter. Sugars are expensive to measure, so we just get soluble solids. We know that correlates with sugar content. We know if soluble solids content's not above 11%, then the squash is not going to have an adequate balance of sugar. So it makes for pretty easy selection. And again, the dry weight in squash usually peaks about 35 days after pollination, as I mentioned before, and it will tend to go down if a squash is harvested, or if the vine is senescing too much, then actually the dry matter will tend to degrade. And there's all these squash have high dry matter and decent quality, but your commercial varieties would be, off the chart. The same thing with sugar. We've selected inbred lines that start degrading starch very early in development. And so by 45 days, they're already reaching soluble solids contents of, 12% or above. And so again, it leads to quality at harvest. What's the problem with that? And I'll probably finish up on this, is that yield goes down as quality goes up. Why is that? Most of you can answer that question. Squash produces, if you have starch, that's the main component, okay? That's the main photosynthetic reserve other than the seed. So if you're getting squash, a fruit with a lot of starch in it, okay, requires a lot more photosynthetic energy than a fruit that has less starch. And so it's not a one-to-one relationship because both fruit have seed in them, and maybe the seed is utilizing about, say, 20 to 25% of the photosynthetic energy is going into seed development, but most of the rest goes into fruit, and particularly starch development. And so if a grower is growing squash for the supermarkets, I can't convince that grower to grow my variety that tastes better if the supermarket, his squash look nice, and they're going into the supermarket and it's what the supermarket wants. Why should he grow a squash that yields, less? And it's a conundrum, and it makes it very difficult. So most of my varieties I'm releasing are going to smaller companies that are willing to market for local markets. They're willing to market high-end squash. I'd like to get a supermarket chain to market squash as a variety, and then they could introduce a variety that has good eating quality. They would increase their sales of squash. It's difficult to do. And I'll just leave you with a message. If you're in a store and you want to get an acorn squash to eat, realize that at 2 weeks after pollination, the ground spot where the squash was lying on the ground is light green because it doesn't have the sun. You know, you need the sunlight to get the chlorophyll synthesis in the rind. And so if you go into a supermarket and take a look at this is what you often, this is a picture I took in a supermarket. And usually in the winter is when you see it. So when were these squash harvested? Probably between 20 and 25 days after fruit set. 20 to 25 days. They're totally immature. There's literally no sugar in them. They're low dry matter. They're essentially inedible, you know, and yet this is what the stores are. And it's not because the stores don't want quality. It's because they don't know how to evaluate a fruit like acorn. They don't, you know, and you can't converse with them on that. I mean, it's just like hitting your head against the wall. This is interesting because this Big Chewy actually, they deliver one of our major supermarket chains. They're a big grower. I think they have locations in Mexico and other areas. And in the fall, this was harvested in the fall. So their source of squash in the fall, at least when you get this orange ground spot, it means the skin has accumulated carotenoids in it. And it's probably close to at least 40 days after fruit set. So it's got maximum starch content. It might not be too high in sugar, but this squash was actually edible. I won't say it was top of the line, but it was actually edible. And this is what I was going to show you. We do all these cooking tests, and we have tremendous variation in carotenoid content. We've also done carotenoid profiles. We can't do that right at the moment because I don't have access to the lab. But what this profile has allowed us to do, we can identify, for instance, we can identify lines that are high in beta carotene. I can identify them just with the naked eye, and we use this color fan. We only use the last 7 colors. These colors don't correlate with carotenoid content. And we can only do it with Cucurbita Maxima and Maxima. The carotenoids are so low in acorn that we can't use this. But this is very handy. Some of the seed companies I work with use this color fan. It was developed for eggs. And you wonder, well, why would that work for squash? Because eggs contain xanthophylls, hydroxylated carotenoids, which you would think would be more yellowish. But anyway, it works fine. And I'm going to end right there on that show pumpkin. Thank you. If you have questions, sorry, I took too much time.
So do we have any questions? Well, I can start out with a question. Do you guys actually breed for the strength of the stem when you're breeding pumpkins?
Yes.
Oh, okay.
Do I breed for the stem? Yeah, you actually do because it's an important trait. And so we did some research on them. And certain varieties will accumulate lignin and cellulose faster than others. But you have the same problem with breeding for stem strength to some extent as you have for breeding starch content, because the dry matter in the flesh, the starch content in the flesh, it's a storage organ for sugar that feeds into the stem and helps the stem to harden, okay? And what we've found is that varieties that are lowest in dry matter have the weakest stems. So if you raise the dry matter in a pumpkin to get better stem strength, you tend to lower the yield a lot. But we've gotten around that to some extent. But I mean, that's a good question. And I might add that in these show pumpkins, there's fanatical growers. There's an association in New Hampshire, and I've spoke to them a couple of times. Well, they want answers, you know, how can I get the largest pumpkin? And just the physiology of why these pumpkins grow big, and just the physiology of growth in itself is very complex, you know, in terms of what's going on in cell division and cell size and so forth. So I have not given them, I've learned more from these growers than they've learned from me. But anyway, it's very interesting. And they're always interested, oh, is there some growth regulators I can spray on these and get them bigger and so forth and so on.
Any other questions?
Is there a correlation between storage and starch and sugar content?
That's a good question. Did you hear the question? Is there a correlation between storage and starch and sugar content? So what happens, you know, pumpkins respire. You know, when you harvest a pumpkin or squash, it respires. So you put a squash in storage. For one thing, the carotenoid content tends to go up, particularly in the machadas. Will increase. It depends on the variety, but it increases between 3 weeks to 8 weeks, and then peaks. So actually, the vitamin content actually tends to increase in not so much in acorn, but in kabocha. And the other thing that happens is the starch breaks down into sugar. You have respiration, so the starch breaks down, and you tend to get a little higher sugar in storage. So the breakdown of starch is usually proceeds faster than the respiration, so you tend to get higher sugar. And like in the butternuts, sometimes they have to be stored for some time in order to get the sugar content up where it balances with the starch. And what they found in studies, and we've followed up on this, New Zealand's done the most thorough work on squash because it's a major export item, and the government actually funds applied work on squash. And what they showed is that they could take squash and they could treat it with high temperature, like 30 degrees C, for 5 or 6 days, and that accelerated the enzymes which break down starch, and then they could take that squash and put it back in the preferred storage temperature, say 10 degrees centigrade, and the degradation would continue, because when they ship their squash to Japan, when it reaches Japan, it's got to be a minimum of 20% dry matter and a minimum of 11% soluble solids. So that work is important. So now when my squash comes into the greenhouse, I tell the greenhouse manager, I said, bump up the temperature for a week, you know, because I have to get the sugar content up in my machadas, because I'm working with a company in New Zealand. I got to send seed to them, so I got to do cooking tests on those squash, and so that's an important thing to understand. Now, it used to be that they did that to all squash, and they do it with sweet potato. Is you cure them. It turns out that the squash don't have to be cured for disease or anything like that, because, you know, at 55, 60 degrees, you know, they can actively get corky layers to prevent disease. There's really not much different. But it turns out if you want to sell your squash, I tell growers, if you want to sell your butternut squash, and you're growing the Waltham variety, which happens to be very low in sugar at harvest, the most popular variety has the lowest sugar at harvest of practically any variety I've looked at. And so I say, well, bump up the temperature, you know, when you put them in the greenhouse for a week or whatever, and bump up the temperature, and at least you can start marketing them within 3 or 4 weeks, and the customers are going to be satisfied. If you eat a squash that doesn't have a balance of sugar in it, even if it's fairly low dry matter, it's like eating a dog bone. You know, it's just not good. So that was a good question.
I think I have time for one more question. Yes.
Yes.
How much is the carotenoid content influenced by environmental stress?
Oh, that's a question I probably can't answer. The question was, how much does environmental stress affect carotenoid content? I guess it would depend on when an environmental stress occurred, but early on, I would say you don't need that much sugar, you know, as a substrate for carotenoids, because the total content in micrograms compared to sugar content. So, you know, some of the highest carotenoid contents are in fruit with only 6% dry matter, the Dickinson field type. But I would think environmental stress carotenoid content, but I can't. I don't have any direct information on that. So, what environmental depends on the environmental stress you're talking about. If you leave your leaf area, you know, your starch content is going to go down. I don't know that that would affect carotenoid content that much, to be honest with you. Since you can get it in storage, you know, without. The other thing I might add is once the squash comes off the vine or the vine dies, it turns out the respiration bumps up about double. No one's ever looked into this. This was work done again in New Zealand, but it's something to think about. You know, if you have a healthy plant on the vine, you want to keep your respiration rate lower, keep it on the vine as long as you can.
Okay, thank you very much.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.